Hello students, welcome to EPG Pathshala. I am Dr. P.M. Reddy from the Energy and Resources Institute. Today we are going to talk on module aspects related to commercial release of transgenic crops, environmental and biosafety issues and regulatory considerations for modern biotechnology under the paper Plant Biotechnology and Crop Improvement. Learning objectives of this module are transgenic crops, Indian and global scenarios, concerns pertaining to effects of transgenic crops on human health and environment, research and development and commercialization of transgenic crops, and regulatory system for release of GM crops and products. Let's start with the transgenic crops, Indian and global developments. Before we talk about biosafety issues and other things, we should first know what are the transgenic plants. A transgenic plant contains a gene or genes which have been artificially inserted instead of plant acquiring them through pollination. In transgenic crops, the DNA of plant is modified using genetic engineering techniques. In most cases, the aim is to introduce a new trait to the plant which does not occur naturally in the species. The inserted gene sequence, known as transgene, may come from another unrelated plant or from a completely different species. The inserted gene can come from species within the same kingdom, that is plant to plant, or between kingdoms, that is from bacteria to plant, from animals to plant, and so on. And the terminology of transgenic plants normally, and as we speak, is termed as transgenic plants or genetically modified plants or genetically modified crop. Transgenic plant development. In my earlier lecture, I already mentioned how to develop a transgenic plant. Briefly, for the benefit of this particular lecture, I would like to explain is a gene is isolated. In this case, what is shown here is a gene from bacteria. That is, as DNA is extracted, a particular gene is cloned, and the cloned gene is transferred to binary vector and put it in agrobacterium. This agrobacterium co-cultivated with the plant callus cells, and during co-cultivation, tDNA from binary vector is transferred into some of the cells present in plant calli, and this transformed calli is put in the plant tissue culture medium under selection. Since the tDNA contains antibiotic resistant gene as well, it can be kanamycin or can be hygromycin, etc. This medium where you culture these transformed cells will contain hygromycin or kanamycin, and these only the cells which are transformed with the tDNA containing marker genes are able to proliferate in the culture, and those cells, calli, can be regenerated in plant, and transgenic plants are produced for breeding purposes, multiplication and breeding purposes. There are different stages of GM crop production, in other words, transgenic plants. These stages include identification of genes with a desired trait, clone the gene into a vector, make copies of it, transfer to plant tissues, regenerate plants, then analyze these putative transgenics for the presence of transgenes and test for its safety and other lab analysis and the development of variety. And these transgenic plants, the conformed plants, can be taken to field tests, and if they are safe, are subject to several different regulations. And finally, if government approves, the plants or crop can be commercialized. And after commercialization, when it's planted in the farmer's field, still it will be continuously monitored for efficacy and safety. This whole process takes about 7 to 8 years. There are several different types of transgenic crops are produced with maybe more than 100 different types now, containing various different genes, producing different types of traits. Some of them are given here. There's this development with objectives, integrated pest management, induction of transgenic plants with the herbicide tolerance or resistance, or nutritional enhancement, increase the product quality, or increase yield, or improve stress tolerance, or induce stress tolerance trait into the plant, and plant-based pharmaceuticals. And several other objectives can be listed in this. Different types of plants can be produced. This slide gives a few examples of what are the genes transferred to produce a particular trait in transgenic plants, various transgenic plants. I would like to draw your attention to few genes here: CRY1A, CRY2A, CRY1AC, and so on. These genes and several more coming from Agrobacterium Bacillus thuringiensis. These are beta genes. These are the genes which are transferred to the plant to develop insect-resistant plants, because these toxins produced by the genes are toxic to insects. And another example is a DREB-A gene. It's a transcription factor derived from plant. This induces, when expressed in the plant, induces drought tolerance. There are several other traits. For improving the nutritional quality and the fortification with vitamin A, a complete pathway is incorporated into rice to produce golden rice. This rice in the seeds produces beta-carotene, a precursor of vitamin A. Various transgenic plants are globally cultivated in several countries, about nearly 18 or 20 different countries. Most transgenic crops area which is covered is in the Americas, North and South Americas. In North America, United States is leading with almost 70 million hectares of land is under cultivation of transgenic soybean, maize, and so on, followed by Brazil, 30 million hectares, Argentina, about 24 million hectares, and Canada, 10 million hectares. And other plants in other countries in Africa, Asia, Australia, these are cultivated to a lesser extent, hectares-wise. In India, you can see about 10.5 million hectares is under cultivation with the transgenic crops, mainly of cotton crop. It's the only crop which is allowed to be released in India. Global area of these transgenic crops, data taken up to 2012 indicates cultivation of the area, global area, is continuously increasing with the transgenic crops. You can see here in this same graph, in the developed countries, the production of the area covered by transgenic crops is now getting flattened after 2009 onwards, whereas in developing countries, it's going on increasing. In other words, more and more developed countries are adopting transgenic crops for cultivation. In case of India, the only transgenic crop which has been approved and released is cotton. It is widely cultivated in some states like Maharashtra, Andhra Pradesh, Gujarat, and to some extent in Madhya Pradesh, Punjab, Haryana, Orissa, so on. Several different types of transgenic rice transformed with the Bt genes are being cultivated here. These are insect-resistant cotton plants. Transgenic crops, concerns and controversies. The public controversy normally revolves around why should we develop transgenic plants? Should we release them into the environment? Are the transgenic plants safe? Are transgenics a threat to non-transgenic production systems? Are transgenics a threat to natural ecosystem? These are all different controversies which have been generated, and people try to, scientists as well as public and regulators try to address them. Normally, when you talk about risk, These risks are concerned with human health and environment. Health considerations are alterations in nutrition profile of the food after transgene introduction, introduction of toxins, introduction of existing or new allergens, whether they create any gastrointestinal problems, decreased body weight, toxic effects on liver, kidneys, etc. This has been shown using BT potato, transgenic crop. Lower fertility and reduced birth weight shown by one or two groups with when used BT maize kernels as a food component and compromises immune system was also noticed. These are all related to BT gene. Environmental concerns are concerned with mostly horizontal gene transfer, whether genes transferred from one plant to a wild species, effect on non-target organisms, development of resistance by pests against these transgenic products, harmful environmental effects, unexpected changes in plant physiology. And societal concerns, mostly people are not familiar with the technology. That's why a lot of concerns are being generated. Lack of reliable information, negative media opinion, opposition by the activist groups, mistrust of the industry, particularly multinational companies. Wide public consultations in the country brought several concerns to the fore. The concerns are mainly related to biodiversity issues, health, safety, multinational corporations' stronghold, loopholes in the regulatory regime. An almost unprecedented amount of discussion has been stimulated on merits and demerits of genetic engineering of crop plants. The arguments for and against transgenics are invariably based on visions of the new technology from widely different ethical perspectives. Research and development and commercialization of transgenic crops. For release of commercialization of transgenic crops, they are subjected to rigorous regulatory regime to ensure safe access to products and technologies developed in the country or elsewhere, and provides a level of public confidence that products placed on the market have been assessed as safe. This is the ultimate goal. They should be safe to environment and a human and animal health. That is the ultimate goal of different regulations. The regulatory structure is very tedious in case of genetically engineered crop plants. If you see in this slide, on the right side, the crop plants developed through plant breeding or a traditional technology, the clearance can be obtained. Regulation is very simple. Clearance can be obtained very easily from the government. Whereas genetically engineered transgenic crops, they have to pass through various rigorous tests and regulations, and it would take an enormous amount of time, cost, and energy to show that the plants which are produced are safe for consumption and as well as the environment. So the regulation structure is very rigorous. Regulatory initiatives relating to biotechnology-derived plants are mainly focused on environmental impact and the general concept of safety of the food and byproducts derived from such plants. A sound regulatory regime should be neutral, neither facilitating nor restricting the approval of products or byproducts derived from biotechnology-derived plants or animals. Before new biotechnology products enter the market, it is essential that such products be evaluated rigorously by appropriate regulatory agencies to ensure their safety and efficacy. Regulatory initiatives must be founded on principles that encompass careful evaluation and monitoring, Regulations and standards for determining the responsible use of plant biotechnology in food and agriculture are based on principles that take into account criteria such as benefits and risks, the scientific basis of biotechnology, and the effects on environment, human health, welfare, and the societal acceptability and economic returns. What are the biosafety elements which are to be considered? Nations assess the safety of GM crops almost similarly, various nations. Two major concerns are concerned with food and feed safety, and other one is environmental safety. In food and feed safety considerations, several aspects are being tested. In the transgenic plants, for their toxicity, if there are any toxins present, allergenicity, if they produce any allergic compounds, or nutrition compound by insertion of any gene, whether nutrition quality of the plant has changed, and several other. For environmental safety, they will assess whether this transgenic has a potential to become a weed, which is unwanted. Impact of gene flow into sexually compatible wild vegetables. That's a transfer of transgene from the transgenic plant to a wild species. Impact of transgenic plants or transgenic plant products on non-target organisms and biodiversity. These are the concerns which are generally observed by different countries. Safety is assessed at different stages during the development and formal use of GM crops, right from lab, where the transgenics products are developed, which are tested in the greenhouse, then in confined field trials, and the full safety assessment, both from the animal and human health and this environment point of views. And once everything is satisfied and is safe to release, then they are released into environment or commercialized. Safety of biotech products is established through various approaches. The safety is judged from the gene and the protein level, the source of the gene, are they coming from pathogenic organisms or benign organisms, molecular characterization of the gene, and copy number of the gene in the plant when it's transferred, and the protein's history, whether it's safe to use and consumption, and the function and specificity and a mode of action of protein. For example, Bt protein, what is its function, what is the mode of action on insects, and the level of protein produced, and the toxicology and allergenicity of protein, all these being tested. The crop safety, crop characteristics, we should not, or transgenic plants should not have a different morphology or yield. In a sense, yield can be increased, but it should not go down. That will defeat the purpose. Feed and feed composition, proximate analysis, the nutrient composition is analyzed, and the anti-nutrient composition is analyzed. And all these things will form a basis to say whether transgenic plants are safe or not. Recommendations for staged completion of specific information and data requirements for safety assessment of genetically engineered plants. These are the recommendations made by our government, various bodies of the government. These studies include basically on the two different aspects: food and feed safety assessment and environmental risk assessment. Both these assessments, various studies need to be completed for their acute oral safety, pepsin digestibility, and livestock feeding study, molecular characterization, inheritance, and so on. Several factors which are listed here are being studied, and these studies are conducted either in the field studies or in the non-field studies in the laboratory, or some of them are conducted in both places. So these are the different parameters these are the guide with how the test should be confirmed. These are parameters which need to be tested. There is a partial list is given here. Several other things can be added to this, which are generally assessed. complexity with regard to development and commercialization of cotton transgenics in India. You can see the magnitude of different acts and rules involved in this, right from the basic research till its development and commercial release into the environment. Right from the beginning, for example, to the end commercial release, it is governed by Environmental Protection Act, Patent Act, and from development to commercialization is governed by Industry Act, and also it is governed by Plant Quarantine Order, Destructive Assessment for the Act for the Destructive Insects and Pests, Insecticides Act. Several other acts are taken into consideration, many other acts, and whether the scientists and the regulators they fulfill all these acts and consider these transgenic plants or the method of developing does not violate any of these acts which are in force. What should be to develop a safe transgenic, we should have a perfect or a very good regulatory regime, and the policy need to be formulated based on that, the policy of the country, policy proposed and the field reality, what it is. Professor Rama Swaminathan, father of Green Revolution in India, he suggested certain points which can be incorporated into policy. He says, I quote, "The bottom line of a national agriculture biotechnology policy should be the economic well-being of farm families, food security of the nation, health security of the consumer, protection of environment, biosafety of country, and security of our national and international trade." All these ingredients should be incorporated, according to the eminent scientists, into the policy proposal of our government. To regulate release of development and release of transgenic crops, a National Biotechnology Regulatory Authority is formed under Government of India. It is being the nodal agency, monitored by Department of Science and Technology, Department of Biotechnology. This has earlier had several meetings. Different modalities were concerned. A national biotechnology development strategy has been developed in 2007 under this. National Biotechnology Regulatory Authority. What is it actually? In brief, it's called NBRA. Is an independent, autonomous statutory agency established by the Government of India to safeguard the health and safety of people of India and to protect the environment by identifying risks posed by or as a result of modern biotechnology and managing those risks through regulating the safe development and deployment of biotechnology products and processes. This is the function of this particular regulatory authority. NBRA was set up under DPT to provide a single window mechanism for genetically modified or engineered products or processes. They assist all the things. National Biotechnology Regulatory Authority, abbreviation NBRA, it consists of different committees. Most important thing are Review Committee on Genetic Manipulation, RCGM, which regulates recombinant DNA safety guidelines and regulations. Genetic Engineering Approval Committee, GEAC, is apex body constituted in the Ministry of Environment and Forests under rules for manufacture, use, import, and export and storage of hazardous microorganisms, genetically engineered organisms, or cells under the Environment Protection Act 1986. GEIA approval for import of any GM product used for industrial production, environmental use, or field application. RCGM approval is required for import of GM products for research and development purpose, and GA approval is necessary for commercialization of GM products by companies and institutions. The rules of 1989 define 5 competent authorities which regulate the transgenic organisms. The Review Committee of Genetic Manipulation, or CGM, Genetic Engineering Approval Committee, GEAC, Institutional Biosafety Committee, IBSC, State Biotechnology Coordination Committee, SBCC, and District Level Committee, DLC, for handling of various aspects of the rules. This slide briefly explains the current regulatory system in India. There are various regulatory systems, bodies which regulate the research, development, release of transgenic plants. One is, the most important thing is Review Committee on Genetic Manipulation, RCGM, which is the one solely responsible for research and development and safety assessment. Another body is Genetic Engineering Approval Committee, GEAC. This body responsibility is commercialization of GM products by companies and institutes. This body gives the approval for commercialization. Besides these 2 important bodies, there are several other committees like Institutional Biosafety Committee, IBSC, and the State Biotechnology Coordination Committee, and at the district level committee, all these things. When the transgenic plants are developed, they pass through all these different committees. And one GAAC approves the release of the transgenic crop. Now, this being a central government body under NBRA, it will go to the State Biotechnology Coordination Committee, which can regulate its release or non-release into their respective states for cultivation. And also this will be considered at the district level committees for biosafety. So there are a very elaborate regulatory regime which is being developed to monitor for the development and monitor the transgenic technology and release of transgenic plants. There are other advisory bodies to this NBRA. That is, one advisory body which is important is Interministerial Advisory Board. This is to promote and ensure interministerial coordination with regard to implementation of regulatory system. The board will include high-level representations from key line ministries like environment, forestry, agriculture, and so on. And another advisory body is National Biotechnology Advisory Council to address overarching policy-related issues that may affect regulation of biotechnology in India. The council members include representatives from scientific community, farmers, consumers, public and private sector, and civil society. All these advisory bodies and finally, which come under National Biotechnology Regulatory Authority and various regulatory regimes, they are the ones screen these transgenic plants for their safety, for human health, animal health, and environmental health. When it is approved, they are allowed to release into farmers' field. It just shows a workflow diagram representing how an application made by any scientist or a commercial unit which develop a transgenic crop, and how decision is made with regard to that application under NBRA. The various stages are listed here, which I already explained. It passes through several committees and assurance and safety is assessed finally. So the approval committee says whether it is safe, can be released, and it's not safe to be released. That's the final judgment is given to that. Today's lecture, we try to examine what are the real concerns related to transgenic products and how various regulatory bodies are trying to address the problems. Research and commercialization of GMO crops created controversies across the globe. An almost unprecedented amount of discussion has been stimulated on the merits and the demerits of genetic engineering crops and has divided the public and the scientific communities. The arguments for and against the transgenics are invariably based on visions of new technology from widely different ethical perspectives. The need to ensure the biosafety of genetically engineered microorganisms and plants or animals is undoubtedly one of the most critical challenges that the agriculture biotechnology industry and regulatory agencies face. Thank you.